Hello, my name is Mary Fantasia. Today is the 16th of December 2019, and I'm going to be presenting Unit 4, Case Study Number 1 and Case Study Number 2 to you. So we'll go ahead and begin with Case Number 1, in which we are given an infant who was born 24 hours ago. She had a normal birth, but 24 hours after being born, she's developed some symptoms of lethargy, hypothermia, and apnea, and the doctors think she has a metabolic disorder, so they order some labs for her. And we need to distinguish, given these four labs, blood glucose, urinary acids, serum fatty acids, and plasma glutamine, which would be the best lab value to distinguish between a disorder in beta oxidation versus the urea cycle. So first we'll go ahead and review beta oxidation and how blood glucose levels and serum fatty acid levels would be affected if we had a deficiency in beta oxidation. Okay, so real quick, lipolysis is going to free our fats, triglycerides, into triglycerol and free fatty acids. And then those free fatty acids will need to be activated by our first enzyme. Our 16-carbon free fatty acid will be activated by fatty acyl-CoA synthase. And this is going to allow us to go to the next step. So a CoA enzyme is added onto the 16-carbon fatty acid, and this requires ATP. And so after this, with fatty acyl-CoA synthetase, we form fatty acyl-CoA. And this fatty acyl-CoA is able to be combined with carnitine, and a CoA is then kicked off with the use of our enzyme, carnitine palmitoyl transferase number 1. So now we have fatty acylcarnitine formed, and this is just for transport across the mitochondrial matrix, because right now we're in the cytosolic side, and beta oxidation occurs in the mitochondrial matrix. So we need to transport our fatty acyl to the right side. Okay, so our fatty acylcarnitine is able to move through the translocase into the mitochondrial matrix. And then our second enzyme, carnitine palmitoyl transferase number 2, it's going to, we're going to kick off the carnitine, recycle that, and then we're going to add a CoA so that we have, again, fatty acyl-CoA form, which is now able to undergo beta oxidation. So as you can see here, we have beta oxidation, where our fatty acyl-CoA is then undergoes a series of reactions in which we end up forming FADH2, NADH, and acetyl-CoA. And this FADH2 and NADH is used for the electron transport chain to form energy, and our acetyl-CoA is also used as a substrate for ketogenesis, in which we form ketone bodies, which is an energy source for the brain in fasted state. And we're also going to, that acetyl-CoA is going to activate pyruvate carboxylase, which is an important enzyme in gluconeogenesis. Okay, so usually problems with beta oxidation occur with the transport of that fatty acyl-CoA across the mitochondrial matrix. It happens with our enzymes. Either a dietary deficiency in carnitine can create a problem, or a deficiency in this enzyme here, carnitine palmitoyl transferase 1 or carnitine palmitoyl transferase 2. So if we have a deficiency in either of these enzymes, we're going to have a problem as we're not able to undergo beta oxidation. And as we know, beta oxidation is an important fuel source in the fasted state. So if we're unable to create these products of beta oxidation, we're going to have hypoglycemia. So that is our first lab value, blood glucose. We're going to have hypoglycemia with a deficiency in beta oxidation. However, I think we have better lab values here that we can also consider. Also, serum fatty acids. Serum fatty acids are going to be elevated if there's a deficiency in beta oxidation because they're stuck at this step, the first step. Lipolysis has happened, there's these free fatty acids, and they're unable to undergo beta oxidation. So we have an increase in serum fatty acids. All right, but we'll look at the other lab values and consider these. So now we will look at the urea cycle to look at our other lab values. So the urea cycle is, as we know, important in the body being able to rid ammonium, which is a byproduct of protein catabolism, and it can be toxic to the body. It can cause all kinds of problems, swelling in the brain, So it's really important that the urea cycle is working so that we're able to rid the body of ammonia. So any problems in the urea cycle is going to be detrimental, especially to a newborn. So we'll go ahead and discuss the urea cycle and some of the deficiencies that you can have and the lab values that you're going to see associated with those deficiencies. So with the urea cycle, first we have our enzyme ALT, otherwise known as alanine aminotransferase, and that's going to allow alanine plus an alpha ketoglutarate to form glutamate and pyruvate. And glutamate is very important to us because it's going to carry that ammonia, and it's going to allow us to travel to the liver so that these ammoniums can undergo the urea cycle. Okay? And then glutamate dehydrogenase, our glutamate is going to be, the ammonia is going to be released, and we're going to form alpha ketoglutarate. We're also going to form NADPH, which is important for fatty acid synthesis. As we remember, NADPH is very important. So, here we have the urea cycle drawn out. So, our first enzyme here is carbamoyl phosphate synthetase number 1, and it needs NAG as a cofactor for it. So, a deficiency in NAG or a deficiency in carbamoyl phosphate synthetase is going to be the first enzyme deficiency that we could see. Here we have bicarb with ammonium with ATP forming carbamyl phosphate. And then our second enzyme, ornithine carbamyl transferase, is going to allow carbamyl phosphate to combine with this ornithine to be shuttled out into the mitochondrial matrix, forming citrulline. And then with arganosuccinate synthetase and the addition of aspartate, we're going to form arganosuccinate. We're going to kick off a fumarate with the help of arganosuccinate lyase to form arginine. And arginase, our next enzyme, is going to be responsible for the release of urea, which is a less toxic byproduct of the ammonia. And it's going to travel to the kidneys, and it's going to be excreted into urine. And then it's going to get, again, recycled into ornithine, and this urea cycle is going to continue to happen. So if we have deficiency, let's go ahead and start. We're going to be looking at some values: urine orate, blood citrulline, blood arginine, and blood NH3 ammonia. So with deficiencies, we're going to see elevations in certain parts of this. So for example, if we have a deficiency in carbamyl phosphate synthase number 1, we would have low urine orate levels, low blood citrulline levels, low blood arginine levels, and we would have, in all of these, elevated ammonium levels. And then with a deficiency in arganosuccinate synthetase or arganosuccinate lyase, we would have elevated blood citrulline levels here, and also elevated blood ammonium levels. And then with a deficiency in ornithine carbamyl transferase, with this one we would see elevated urine orate levels and elevated ammonium levels. So for that, yes, it would be good to look at urinary acids for a deficiency in ornithine carbamyl transferase, as we would see elevated urine orate. However, this is just one deficiency in the urea cycle, and it's not accounting for the rest of the deficiencies that could happen in the urea cycle. So that is not the best lab value to look at. Urinary acids, we'll go ahead and cross that out. It's not the best one for this. However, it would help us to determine if we had a deficiency in the ornithine carbamyl transferase. Okay, and then lastly, plasma glutamine. If we had a problem in the urea cycle, in any of these enzymes, we would have elevated plasma glutamine because it would be unable to enter the urea cycle. So that would also be a great lab value to determine, and I think probably the best lab value to determine if this was a disorder in the urea cycle. So I think that plasma glutamine would be the best, along with plasma ammonium, even though it's not an option, would be the best two lab values to determine if we had a deficiency in the urea cycle. Okay, and then lastly, we'll go ahead and go through, so a full blood workup was ordered for this female, and the data is presented below. Her ammonia level was elevated at 314, so that tells us it was a problem in the urea cycle after all. And her urine orotic acid level was elevated at 473, and the plasma citrulline level was normal. So that tells us that it was a deficiency in ornithine carbamyl transferase. And then in this patient where orotic acid levels are elevated, list one urea cycle enzyme that is most likely deficient and one urea cycle enzyme that is least likely to be impaired. So urea cycle. So this one, ornithine carbamyltransferase, is the one that's most likely to be impaired, and the one that's least likely to be impaired would be like arginase. We'll choose arginase. That's the one that's least likely to be impaired because we have this. We would not have elevated levels of urine borate. All right, and then case study number 2, we have a 4-day-old infant, and pretty much we have elevated levels of phenylalanine, and they're given a diagnosis of phenylketonuria. And so as a consequence of this presentation, which amino acid may become essential? So normally we can break down phenylalanine using phenylalanine hydroxylase to form tyrosine. So tyrosine, we don't have to get it from our diet because we can just break down phenylalanine into tyrosine like that. But if we have a problem with breaking down phenylalanine and we have elevated levels of phenylalanine and phenylalanine ketonuria, then we probably have a deficiency with either BH4 tetrahydrobiopterin or phenylalanine hydroxylase. And so we're unable to form that tyrosine. So we're going to get that tyrosine from our diet. So as I said, the potential causes of the fetal ketonuria would be deficiencies in BH4, which is required for this reaction, or phenylalanine hydroxylase. And then we have some tests here, a BH4 loading test, and how would we summarize the results of this test for patient A and patient B? So BH4, as I said, is the required cofactor for metabolism of phenylalanine to tyrosine. And so this first patient, they're both given BH4, and if this patient had deficiency in BH4, then you'd be able to, if we gave them BH4, then their phenylalanine levels should decrease because they're given the cofactor required for the breakdown of phenylalanine. So patient B here has a deficiency in BH4 because with BH4, when they're given it, their levels of PHE drop down over time, whereas patient A must have a deficiency in phenylalanine hydroxylase and not BH4 because with BH4, their levels of phenylalanine remain the same and actually slowly elevate, meaning that they don't have a deficiency in BH4. Okay, thank you very much.